Now we have coronavirus. If we were, we didn't have the variation, coronavirus would just wipe us all out. Basically, there is no immune response that would be able to prevent it if it was we were all the same 100%. So it gives you variation. There's a population, I think in Africa, that they don't get HIV even if they're injected with HIV virus, and that's maybe because there's something different in their DNA that prevents it. So it gives you variation. Getting to definitions, I want you to know the definition of DNA. It stands for deoxyribonucleic acid. If you didn't take biology before, a lot of people just don't know what DNA means. They know there is DNA, but they don't know the whole form. So I want you guys to know that. I might ask you on the exam. The DNA is going to carry the hereditary information that can be passed on from the parent to the offspring. And it's what's guiding the development and functioning. It has 4 base pairs. You don't have to know these today. You will have to know these when we get to the genetics chapter. So this is the model of DNA. It's a double helix model. It's going to have 4 base pairs. It's going to have sugar phosphate backbone. And these 2 people here, they're the ones who discovered it or who got credit for discovering it. That's Watson and Crick. Recently, they started giving credit to Rosalind Franklin. And so basically the procedure they did, it's called X-ray crystallography, that they did to find out the shape of the DNA. They didn't know how to read it. She's the one who read it for them, and she explained it what it is, but she never got credit for it. So right now they started giving her credit for the discovery of DNA as well. So it's 3 people: Rosalind Franklin, and then Watson and Crick. So those 3 people get credit for discovering this. So I want you to notice the diversity. Let me see if I can play this. So you can see there are these coral reefs there. So I don't know if the video is gonna go to some more. So you can see another fish right here. So I just want to show you some environment that has a lot of diversity. So this has a lot of diversity. I don't think the video is gonna show much. Let's see. Okay, there you can see a lot of different types of fish, more coral reefs, and then you also see some space that's kind of completely white. There's some other type of fish. So coral reefs, they are considered very balanced because there is a lot of diversity there. You can see some fish here. So I think I have it on the next chapter, the next picture. So here's a photo of coral reef. And then here is another photo that's not showing much. So this is what I mean by diversity. So it's going to be very balanced environment if there's a lot of different types of organisms that are present there. So when maybe you talk about government employees, they want to hire a lot of people from diverse backgrounds. So that refers to maybe, different types of people. So they want to have a lot of diversity in the department, basically. So that's when you have a lot of different types of organisms here. So that's high biodiversity compared to here. Do you guys know, or can you guys tell if there are any organisms in here? Do you think that there's anything living in this photo? Not visibly, but yes, there's microorganisms that might be there, but still it's a low diversity because there's not a lot of species compared to this environment. So coral reefs are very, very balanced. So the scope of variation among living organisms. So we're gonna switch the gears a little bit. We're gonna get to, we're gonna start dividing some of the organisms into different groups. So if you get a pile of books, you wanna start separating those maybe by fiction or nonfiction first. And then you wanna go ahead and do, cooking books, maybe fitness books or something like that. So you wanna start organizing them. So that's what we're gonna do next. And that's the classification system. So there's 2 major types of organisms. There is prokaryotes and there is eukaryotes. Pro means before. You don't have this on the PDF, so you want to write this down. Pro means before. Karyo means nucleus. which means this is before the nucleus came around. This is without the nucleus. Eukaryotes means with nucleus. Eu means true, karyote means nucleus, so that's with the nucleus. So prokaryotes without nucleus, eukaryotes with nucleus. When you compare these two cells, they differ in size as well. Eukaryotes bigger in size, and prokaryotes are smaller, and they're simpler. You can see there's a lot of structures inside here compared to here, there's not much. And again, I don't want you confusing the nucleus with the chemistry nucleus, because their atoms are found in the nucleus as well. It just means center, basically. So you can see that there is this nucleus, that's where the DNA is found for eukaryotes. For prokaryotes, you can see some circular structure there, but that's not a nucleus. It's just something that's floating around. And you can see that the DNA is just floating around in the cytoplasm, which is this liquid part. So there's nucleus for eukaryotes and there's no nucleus for prokaryotes. I might ask you on the exam, what is one difference between these? You're gonna say prokaryotes don't have a nucleus and eukaryotes have a nucleus. So prokaryotes are single cell organisms in which DNA is not contained in nucleus. So it's just floating around. This is DNA, it's floating around. There's nothing around it except the cell wall, and that's the whole cell that's there. For this, I want you to memorize 3 shapes. If it's in circles and it's a grape-like clusters, it's called cocci. So it's cocci if it's circular. If it's in the rods, it's called bacilli. If it's in spirals, it's called spirochetes. That's all I want you to know. You don't have to memorize the pneumococci, streptococci, pseudomonas, or salmonella. That's for microbiology, but it's just showing different shapes. So I want you to know cocci again, circular shape. Rods is bacilli. Spirochetes are spirals. I might give you a photo on the exam and ask you which type is, which. So like I said, if you get a pile of books, first you want to maybe do fiction and non-fiction and separate those. So that's maybe prokaryotes and eukaryotes. Now you want to go maybe in prokaryotes and maybe separate them a little bit more. We have 2 types of prokaryotes. We have bacterium, and this bacteria, and then we have archaea. If you think about the word archaeology, that's study of old things. They named it archaea because these type of organisms, the type of environment they live in is very similar to old Earth, which means they are found in really, really hot places in volcanoes maybe, or they're found in really cold places, or they're found in very salty environments. So they are basically, their environment they live in is very similar to old Earth, so they're called archaea. So bacterium is a member of prokaryote domain bacteria, and we have 2 domains basically. So we have bacteria and archaea. Those 2 are prokaryotes, and then we have eukarya. Archaea, it's more closely related to eukarya. It comes under prokaryotes, but there are some characteristics that make it closely related to eukarya. So actually, before I start getting into a little bit more detail for these groups, I think we will do that next time. So I'm going to stop here, but before I do that, I want to know if you guys have any questions. You guys have any?
Can you please go to the last slide? I didn't finish writing quite everything down. I'm sorry.
This?
Thank you. Yes, thank you.
These slides are available on Canvas.
Okay.
And the questions are not on there because I had the questions right before the lecture. So if you like having questions, then you might want to write them down, but all the other slides are there.
Thank you.
No questions on any of the slides? No? Okay, let me stop the recording.